All right, everyone. Welcome to FSN 328 and our YouTube. So let's start off with sketching out that diagram like we started last time, right? Where we had the endothelial cells and all that. And we're talking about this part. So we're on pathway number 1 with chylomicrons. So we're going to go through and we're going to talk about how dietary lipids end up in different tissues after we eat them. So remember, the lipids are packaged in enterocytes. And so we've essentially left the small intestine, and then they've gone through the lymph, and then they end up in these capillaries. Okay, so what are these? These are endothelial cells, so we're saying this is the blood. And I probably put this here, right? And we said that this is lipoprotein lipase, that goalpost structure. Okay, and we said this is a chylomicron. It has ApoC2 is one of its apolipoproteins, and the other one is what? Did I tell you that yet? ApoB48. So when you look at the structure of chylomicrons, if we say this is the generic structure of all the lipoproteins, if this is a chylomicron, this protein, this kind of integral membrane protein, is ApoB48. Remember the relationship between these guys, B48 and B100? Yeah, size. B48 is called that because it's 48% the size of B100. And next quarter in lab, I'll tell you how that works. They come from the same gene, same mRNA, and a modification in the mRNA yields 2 different proteins. B48 is a large protein, wraps around a chylomicron. It's structural. So that's what this is. We're going to say this is B48. ApoC2 is another protein there because ApoC2 activates lipoprotein lipase. You have that in your notes, right? ApoC2 activates lipoprotein lipase. ApoC2 activates lipoprotein lipase, LPL. Do you have in your notes what LPL does? Yeah, so LPL, this guy, hydrolyzes triglycerides to glycerol plus fatty acids. So this is LPL. So this enzyme is attached to the endothelial cells, binds ApoC2 and its chylomicron, and starts breaking down the triglyceride that's in there. Where'd that triglyceride come from? Yeah, whatever you just ate. So this is in the next, so this is in the previous meal. That's where this triglyceride came from. Went through the enterocytes, packaged into chylomicron, through the lymph, and now we're following it into the blood. Okay, so that's one thing that's in here. It's triglyceride. What else is in that chylomicron? Fat-soluble vitamins, good. What else? Cholesterol. So we're going to say this is cholesterol. It's also in there. LPL, though, is basically breaking down triglycerides so that we end up with fatty acids plus glycerol that are made into a triglyceride. And so let's say this is a cell in the underlying tissue, which means this is probably a fat cell. Or this is mammary tissue, or this is muscle tissue. Most of that dietary triglyceride is going to end up in fat cells. If you're a guy, we tend to express more lipoprotein lipase around our abdomen than women. That's one of the reasons we store more body fat around our abdomen. Women tend to store it in other places. Usually it's a little bit lower around the hips, something like that. But that's one of the reasons is that lipoprotein lipase in that fat tissue has a higher Km than the lipoprotein lipase anywhere else. And you don't have to remember that. Just remember, lipoprotein lipase is most active in fat tissue. It's one of the reasons we're good at storing dietary triglyceride in adipose tissue. Some of it ends up in the other tissues, but most of it is in adipose tissue. Okay? And that's how it gets there. Now, if we go through this process, when approximately 80% of the dietary triglyceride is hydrolyzed, ApoC2 releases. So when about 80% of the triglyceride is hydrolyzed, ApoC2 dissociates from lipoprotein lipase. So most of the triglyceride's gone. Cholesterol's left. B48's left. Now we call this particle a chylomicron remnant. Now we're here, chylomicron remnant.
ApoC2 dissociates from, what was it?
Comes off the chylomicron. Cholesterol is transported to the liver. Where'd that cholesterol come from? The diet, from the meal. Got all that? So for example, if I ask you to describe how dietary lipid is transported from the gut to storage tissues, account for triglycerides and cholesterol as part of this process, include your lipoproteins, your apolipoproteins, and any enzymes, you're going to basically draw this out and describe it. I think I have everything there.
Is the apolipoprotein B48 still with it when it goes to the liver?
Yeah.
When C2 dissociates.
it's the chylomicron. Yeah. C2 can now bind to another lipoprotein. C2 kind of floats from lipoprotein to lipoprotein. It's promiscuous. Okay. B48 stays there. It's part of that lipoprotein. Good there? Now you know a lot of information that's going to help you in the next pathway. So we're going to call that pathway number 1, and I'll give you more context for that one or applications in a few minutes.
Just so C2's job is just to like help hydrolyze the triglyceride inside.
So C2's major function activates LPL. Yes.
In addition to activating it, it also like binds it to LPL, like helps it bind to LPL.
Yeah, so it's got a binding site for that enzyme that probably causes a conformational shift, activates the enzyme. Mm-hmm. Right, so imagine though when that lipoprotein is getting smaller, as it becomes smaller, C2 is pushed off. So it just releases. So it's kind of a size effect on when C2 actually releases from that. And this one might be helpful. So the summary. Chylomicrons deliver fatty acids attached to triglycerides derived from dietary fat to non-hepatic tissues, mostly muscle and adipose tissue. Remnants, chylomicron remnants, deliver dietary cholesterol to the liver. So that's how it gets to the liver. And now we can look through the other ones. The next one we're going to talk about is Vldl and Ldl. So the main function of Vldl is to deliver fatty acids attached to triglyceride derived from liver synthesis to non-hepatic tissues like muscle and adipose. And then Ldl are made from Vldl. And Ldl deliver cholesterol derived from liver synthesis to various tissues. So now we're in the two lipoproteins that transport endogenous lipids. So let's talk about Vldl and Ldl next. Pathway 2. This is a Vldl particle or lipoprotein. This one has ApoB100 in humans. And that's what we're talking about. You remember that bar graph I showed you last time? What's the major lipid in Vldl? Vldl chylomicrons are triglyceride rich. So mostly triglyceride. Where did most of that triglyceride come from? Synthesis in the liver. So a couple times we've said, well, if a person consumes a lot of calories and their liver has adequate energy stores, adequate energy status, they'll take some of those carbons from those extra calories, convert them to fatty acids, like palmitate, make triglycerides, and then export it in the blood. We've mentioned that a couple times. So now what we're going to do is we're going to take that process and add more detail to it. So most of these lipids in the form of triglycerides and cholesterol were made by those liver cells. They put it in Vldl. So in other words, they wrap it in a lipoprotein that's got phospholipids. Around it, lipid core, apolipoprotein B100, and that Vldl goes into the blood. I could actually change this. Maybe this will help. And essentially does the same thing as chylomicrons. So the steps in the process in the capillaries is the same for chylomicrons and VLDL. So in other words, this could be a VLDL. If it's a VLDL, it's got B100. And you can decide if you want to make yours messy like mine or if you want to write it separately. But if this is a VLDL, it's B100. But the VLDL also has C2. So as that VLDL goes through the blood, it picks up C2. And the C2 allows it to interact with lipoprotein lipase. Same thing happens. Triglycerides are hydrolyzed, they're stored, goes through this process. But now rather than a chylomicron remnant, what would we call this one? The VLDL remnant, aka IDL. Same thing. VLDL remnant or IDL. So we can say this is, how you want to call it, VLDL remnant. Except obviously it's going to have B100. So now the VLDL remnant leaves lipoprotein lipase, goes through the blood. Here's where they're different though. Here's our VLDL remnants. There's two forks in their road. Here's number one. VLDL remnants can either be taken up by the liver. So let's say this is a VLDL remnant. It can be taken up by the liver or become LDL. There's no C2 left, but is there B100? There is B100. So the only apolipoprotein in LDL is ApoB100. What lipid does it have? Cholesterol. We've all heard about LDL, and we say, oh, LDL is the bad cholesterol. Why? Because this is the particle that can be involved in atherosclerosis, in heart disease. Where did it come from? VLDL. Where did its cholesterol come from? The liver. Okay? So we'll talk about that in a second. Now, about half of the VLDL remnants are cleared by the liver. About half of the VLDL remnants become LDL. Okay, now look at LDL. LDL also has kind of two pathways or options. Some LDL are taken up by the liver. Some LDL are absorbed by extrahepatic tissues. So again, their major lipid is cholesterol. And in fact, the major function of LDL is to transport cholesterol to extrahepatic tissues. So that's good. Cells throughout the body need cholesterol. If they can't make enough, they can pull cholesterol from the blood. They just get it from LDL. So what we'll talk about in a few minutes is cells can actually just make receptors and pull LDL from the blood to get more cholesterol. If they need to make more membranes, or if they need to make more steroid-type molecules from cholesterol, that's their resource. That's their substrate. They just get LDL. The problem is, some of this, we're going to say, including the intima of coronary arteries. So it's that process there that gives LDL the risk of being related to heart disease. Its major function is absolutely essential. One of the artifacts, though, if a person has too much LDL cholesterol, it's more likely to be deposited in the arterial walls of coronary vessels, and that can cause atherosclerosis. That's one of the things that can cause it. Doesn't mean if a person has high LDL cholesterol, they're going to have heart disease. It means that it's increased risk. That's all. Percentages here. Most LDL, about 75% of it, is actually taken up by the liver. About 25% of it is delivered to extrahepatic tissues. So it's pathway number 2, VLDL, LDL. So if I ask you, tell me how a person that's consuming too many calories, including too many carbohydrates or too much dietary protein, can actually end up adding adipose tissue around their waistline. And if you include lipoproteins, you're going to talk about VLDL, and that's it. Right? We didn't talk about chylomicrons because I said protein or carbohydrate. They have to go to the liver to be converted. So the liver can convert some of the macronutrients to triglycerides and then export it in VLDL. And again, if we kind of go back to our conversation before, we said, well, why are fat-soluble molecules put into chylomicrons? Well, it's so the liver can avoid accumulating lipids. It's kind of the same thing here. If a person's consuming excess amount of calories and macronutrients, the liver can convert it to fat, and the liver will try to get rid of it. So the liver will put it in VLDL, send it to extrahepatic tissues. So in this case, think about it. If I'm consuming significant amounts of protein and too many calories, some of those amino acids in the liver can actually be converted to fatty acids. And we'll talk about that next lecture. And then those fatty acids can be converted to triglyceride and put into VLDL. So the liver has to get rid of those amino acids somehow. If it doesn't need them, it can convert them to fat, put it into VLDL, because now what can VLDL do? Put it in adipose tissue. And we have the ability to store significant amounts of triglycerides. So in that case, essentially what we've done is we've taken the excess macronutrients and stored it as fat. Okay? So it's important that you understand these pathways and their context. Like, why are they important? So as you go through this, I would say practice one at a time, sketch it out, write in all your details, make sure you have everything, and then think about what are the important scenarios that involve that lipoprotein. Okay? And again, I can give you some study stuff as well if you like. That one's probably the more complex one, VLDL, LDL. HDL is pretty easy. We'll go through that in a second. HDL, high-density lipoproteins, collect or scavenge cholesterol from non-hepatic tissues and deliver it to the liver for excretion. So you can look at it like this. VLDL are taking cholesterol from the liver. HDL are taking cholesterol from here to the liver. They pick up extra cholesterol from tissues. And we'll go through the HDL cycle in a second. I think the next few slides kind of summarize stuff on here. So this one says the functions of apolipoproteins. We've talked about a few of them. They're a structural component for the synthesis and stability of the particle. Who can give me an example of that? Which apolipoproteins? Structural. B48 and B100. Good. They provide recognition sites for cell surface receptors. We'll go through that in a second. The third one, they act as cofactors for enzymes involved in the metabolism of lipoproteins, like ApoC2. It activates lipoprotein lipase. So it's a cofactor. So apolipoproteins help determine the fate and function of each type of lipoprotein. A good example of that is now let's look at number 2. Some apolipoproteins provide recognition sites for cell surface receptors. Apolipoproteins bind to the receptors on cells, and the lipoproteins are removed from the plasma or blood by receptor-mediated endocytosis. We're going to talk about LDL and how that works in a second. Okay, let's look at the next one. So this is a table that you can cross out that molecular mass. I know, fascinating, but I won't ask you this. But B100 is one of the biggest proteins in the body. And that's another reason LDL are unique, because they have that one. And then we have A1 and C2. You can see are pretty small, but B48 and B100 are really large proteins. And you can see what they do. A1 we'll talk about when we talk about HDL in a second. They actually activate an enzyme called LCAT. And they're a ligand, also a ligand for the HDL receptor. B48, we looked at that one. B100 is ligand for the LDL receptor. And B100 also is made in the liver in humans. And in the lab, we'll talk about why some animal species are susceptible to heart disease and some of them are not. We happen to be on kind of the short end of that one. We're very susceptible to heart disease. One of the reasons, because our LDL have B100, kind of sets us up for heart disease. We'll talk about mechanisms in 329 lab. And then the last one is C2. We talked about that. This is a table to help you if you're drawing out your diagram so you know which apolipoproteins go with which lipoprotein. And here's fun facts about VLDL. Synthesized in the liver contain B100. I'll let you go through those. Here's the lipolysis cascade. VLDL become IDL, then some become LDL. This is probably going to be your favorite one. All LDL are from VLDL via IDL, but not all VLDL become LDL, right? I mean, that's just straightforward. Those guys are nuts when they called these that. Huh? It should have said apolipoprotein or lipoprotein good or bad or the cousin or something. As VLDL triglycerides are removed, ApoC2 dissociates. I went through that. LDL particles. They transport cholesterol to extrahepatic tissues, so they're kind of referred to as the bad cholesterol of the blood because they can deposit cholesterol in coronary arterial walls. The only protein component is B100. They lose the other. They're cholesterol rich. Here's a big difference. They stay in the blood for 2 or 3 days. So they can have an effect on tissues. Chylomicrons are cleared very quickly. Once chylomicrons hit the blood, within minutes, most of them are gone. VLDL, maybe a few hours, and then they're gone. LDL and HDL stay around for longer periods of time. And this is also a very interesting table. We're going to look at the left half of this now, and in 329 lab, we'll look at the right half of this. Have you ever seen these values? Cholesterol concentrations in the blood? How many of you guys know your blood cholesterol concentration? Okay. These are kind of typical values. This is distribution of triglycerides and cholesterol in the plasma of people that are fasted. If you give a blood sample, most of the time they have you fast, depending on what they're going to measure. The main reason, one of the main reasons you fast is to let your chylomicrons clear. Okay? So if you take a fasted blood sample and you spin down your red blood cells, if you did not coagulate that blood, it is called a plasma sample. It's typically the way it's done in labs. They'll add EDTA or heparin sulfate. They'll stop clotting by removing the calcium, and then they'll spin it. The fraction on top is your plasma. I mentioned this last time. Now you take a sample of that and you analyze it, because that's where the lipoproteins are. They're not down with the red blood cells. If you do a total cholesterol value, this is kind of a typical value, 176. I'd say that's a pretty good average. As a matter of fact, this lab might even be a little bit below that. Every once in a while, I'll measure a quarter, and it's usually around 170 or something like that. That's a good value. You guys are not a random sampling of the population. We're nutrition students and mostly women, so this has affected this. In other words, if we say that's 100%, now that's total. There's different lipoprotein fractions within that plasma sample. There's a little bit of VLDL. There's not much because most of it's cleared. Person fasted, they waited a few hours, chylomicrons are made, they're removed from the blood, VLDL is cleared. At that point, there's not many VLDL being made. IDL even less. Here's LDL. 115 out of that 176 mg per deciliter of cholesterol is in LDL. Humans are what we call, what some people call LDL animals. We carry most of our cholesterol in LDL, the particle that can hurt us. Not uncommon for it to be 2/3 of the cholesterol in a person's blood is LDL cholesterol. Now, as that LDL circulates through the blood, it can deliver cholesterol to tissues, but that LDL can also be altered or damaged and lead to cardiovascular plaque. So the longer it's in the blood, the more likely it is to be altered and be part of that plaque formation. So again, more details on that coming. When you look at that online lecture, I'll give you a couple examples of that. Now look at HDL. That's the second highest fraction in a fasting sample. And this one is about 45 mg per deciliter. Not a bad value. That's probably for men, that's about average, maybe a little above average. For women, that's probably below average. You guys tend to have higher than 45. I've seen these as low as 25. No, actually, yeah, about 23 or 25. If you drop below about 25, 26 on this, that's increased risk, significant increased risk for heart disease, because the HDL is basically taking cholesterol from tissues and getting rid of it. So what do we generally want? High HDL, low LDL. Now, this is pretty common in a healthy female population. These tend to get closer. In some individuals, they're actually flipped. And cardiologists have a term for that. I can't remember. Cool people, or I can't remember what they call that term. But anyway, it's a kind of a unique subset of the population that's protected from cardiovascular events because of that. They have higher HDL than LDL. Okay? So that's one of the things we look at. So remember that. Now, one of the things is, well, how can we actually affect LDL cholesterol in the blood? One is probably choose the right parents. That's genetics. That's kind of part of this. There's things like your dietary habits, exercise, insulin sensitivity. There's quite a few things that can affect LDL cholesterol. And fiber, for example, is one of the things that we're going to talk about in a minute. In order to understand how LDL cholesterol can be affected, years ago there was a couple guys named Brown and Goldstein, two researchers that early in their careers they won a Nobel Prize for this. And they were physicians in New York, I think in the Bronx. One guy was originally from Texas. The other guy was from New York. They had a clinic. They had a health clinic. They saw a case where a child had excessively high amounts of blood cholesterol. It was genetic. So that condition is called familial hypercholesterolemia. Right? It's genetic. High cholesterol in the blood. Familial hypercholesterolemia. So they started trying to figure out why these children had those exceedingly high amounts of cholesterol. They had so much cholesterol in the blood and in their tissues that they actually had kind of bubbles in their skin that you could feel them and you could depress it. It was actually cholesterol deposited. You know, unfortunately, they didn't have a normal lifespan. So it's not that common to have those excessive levels, but the levels of cholesterol in the blood are affected by genetics. So these guys started trying to figure this out. That led them on to this pathway that is shown here. This looks like kind of a funky diagram. This is a very similar diagram to the first one that was published by these two guys, Brown and Goldstein, when they were doing this research. And I saw one of these guys talk at a science conference a few years ago. It was Goldstein. And I remember because I was sitting, before he went up there, I was sitting there, and this is a long time ago, right? I had a BlackBerry, right? And I was sitting there, and I remember this guy sitting a couple chairs away from me, and we were kind of off in the corner just playing around with his BlackBerry. And I could tell he was frustrated because I was always frustrated with my BlackBerry, so I recognized him. I'm going, oh, another poor guy that bought a BlackBerry, right? A couple minutes later, blah, 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 Goldstein, blah, blah, blah. So he gets up and walks out there. I'm going, even Goldstein can't figure out his BlackBerry. It's not that easy. So he walks up there, and these guys are like rock stars of the science world because they were probably early 40s when they won their Nobel Prize. And when I saw him talk, he was probably 70s. And he joked around. He goes, and now this part, I say this every once in a while, but now once we figure this out, I'm going to retire. I'm like, no, because he stayed in this forever. They figured out this pathway and how it's regulated. So this diagram you can put on the top of this. This is LDL receptor mediated endocytosis. LDL receptor mediated endocytosis. Basically this. LDL receptor mediated endocytosis. So this is the pathway. This is how it works. And somewhere in your notes next to this, you can say occurs in almost every tissue, occurs in almost every cell in the body. And so we're talking about this part right here. How these LDL are cleared. Their cargo is cholesterol. Cells can basically determine how many LDL they're going to take up from the blood by regulating their receptor numbers. So an important thing to think about is these receptor numbers are driven by intracellular cholesterol concentrations. LDL receptor numbers are regulated by intracellular cholesterol concentration. Okay, so we'll go through this process. I take 5 to 10 minutes to go through this, so probably a good time to take a break. And then we'll go step by step. I'll give you about 6 or 8 steps in this process. And then we'll talk about how it's regulated by things like diet. Okay, so let's take a break here.
LDL receptor are regulated by intracellular.
Intracellular cholesterol concentration, yep. Okay, so I think it's recording again, fingers crossed. Let's go through the steps on this process. So again, big picture, this is how LDL are taken out of the blood. This occurs all through the body. And so we can see a single cell. Here's the plasma membrane. And we're going to kind of go through these steps. We won't go through 10 steps, but we'll go through kind of this sequence. Here's an LDL molecule in the blood. And it has phospholipids on the outside. ApoB100 protein looks like a little triangle on there. Right? That's their diagram. So remember, B100 is wrapped around this. And this LDL, its major lipid is cholesterol. And more specifically, it's cholesterol ester. But cholesterol ester are both taken into the cell. So on the surface of the cells, and there used to be some good YouTubes on this and stuff. If you look at these, there's actually these little pits or clefts on the inside, on the outside of the cell that has the LDL receptor anchored. So here's the LDL receptor. In order to form this pit and to close off that pit during endocytosis, there's proteins underneath. And those proteins called clathrin, but I don't expect you to remember that. So this ApoB100 binds to the LDL receptor. Okay, so we're going to say kind of step 1. The LDL receptor binds ApoB100 of LDL by a protein-protein interaction. The LDL receptor binds ApoB100 of LDL by a protein-protein interaction. So what does that mean?
The R groups from the protein or the amino acids interact?
Yeah, it's structural. So the structure of the LDL receptor is recognizes and binds part of the structure of B100. So the reason I'm giving you that kind of detail is genetics affect structure. So now imagine I have an LDL receptor that doesn't bind ApoB100 very well, because maybe I have a genetic sequence in the LDL receptor gene that compromises its affinity for B100. What would happen to the LDL?
Bind as tightly.
Stays in the blood. Doesn't bind as well, stays in the blood. So one of the causes of familial hypercholesterolemia is alterations in the LDL receptor gene. What's another potential gene that could be causing that? The ApoB100 gene. Right? Remember the nucleotide sequence of the gene determines the amino acid sequence. That determines the structure and function of a protein. So either one of those genes, alterations in either one of them can cause altered affinity and altered binding or clearance of LDL, so the LDL can stay in the blood longer. So that's some of the genetic anomalies that they found are in those two genes. Most of them are. Okay, so step 2. The the LDL particles are endocytosed. Right? So imagine this particle binding right here, and then the cell engulfing it. So the LDL particles are endocytosed. To form an intracellular vesicle. And that's what's right here. To form an intracellular vesicle. It's endocytosed, and now we have a vesicle. In other words, all the LDL are in these kind of intracellular compartments. So they're enclosed. So they are encapsulated in these little vesicles. At this point, the LDL B100 is still bound to the receptor. Okay? The vesicles are converted to acidic endosomes. The vesicles are converted to acidic endosomes when protons, the vesicles are converted to acidic endosomes by the incorporation of protons. So now protons are pumped in to those vesicles and they become acidic. Did the entire cell become acidic? Nope, so don't say that. Okay, it's just in the endosome. Typically, what's the intracellular pH? About 7. But now the cell drops this pH to about 5. All right, by the by the incorporation of protons.
What's the distinction you just made there? So it's not the whole entire thing that comes
Right, so pH here, 7.
I see.
pH inside here, 5. Right? The drop in pH causes separation of B100 from what? LDL receptor. Okay. Now, the endosome is then separated into parts that contain the receptors from parts that contain LDL. So the endosome is separated into parts that contain the receptors and parts that contain the LDL. The LDL receptor vesicles recycle receptors to the membrane. The vesicles containing the LDL receptors recycle the receptors to the membrane. Funny how appropriate that saying is. A picture's worth 1,000 words. Right, so that's all I'm saying. This scope here recycles back to the membrane. So the same receptor can go through this process multiple times. Goes to cell surface, kind of cycles through, binds LDL if they're present, goes through this process, goes back to the membrane until they're eventually degraded. Okay? So the receptors are kind of in a dynamic flux. And I'll tell you why that's important in a second. But now look what happens to the rest of it. The vesicles containing LDL fuse with a lysosome. So the vesicles containing LDL combine with lysosomes. Remember what's in lysosomes, right? Yeah, digestive enzymes. So lysosomal enzymes degrade ApoB100 to amino acids, cholesterol esters to what? What's a cholesterol ester? Cholesterol plus a fatty acid, right? So the cholesterol esters are converted to cholesterol and fatty acids. If there's cholesterol, it remains cholesterol. But now we have amino acids, cholesterol, fatty acids that are now in the cell. They're in an endosome because they've been, that LDL particle has been dismantled.
Would you say ApoB100?
Is digested to amino acids. So what happens to the cholesterol? The cholesterol can immediately be used or stored in the cell. And we won't go into a lot more detail on that. There's an enzyme called acylcholesterol transferase that's regulated based on this process, and it determines whether or not cholesterol is used or stored as an ester. So the cell is all tightly regulating cholesterol metabolism depending on the needs of the cell. So overall, though, in your notes, now let's put what? Regulation of LDL receptor mediated endocytosis. What? RCE or RE or whatever. However you want to abbreviate that. I write slow, so if you look at my notes, it's all abbreviations and shorthand junk. I'm sure you couldn't read it. But now we're going to talk about how all this is regulated. What did I say earlier? What regulates this process?
Concentration of cholesterol.
Intracellular cholesterol concentration regulates this. So what that means is if a cell has a lot of intracellular cholesterol, it won't take up more. If a cell has low intracellular cholesterol, it can take up more cholesterol from the blood. So how do cells do that? What do they regulate? I said the receptors is dynamic. Their numbers are dynamic and in flux. So a cell can regulate how much cholesterol it absorbs from the blood by regulating LDL receptors. So intracellular cholesterol concentration regulates LDL receptor numbers. So intracellular cholesterol concentration regulates LDL receptor numbers. Add a little bit more to that by a transcriptional mechanism. What does transcriptional mean? DNA to RNA. Right? It's transcriptional. So in other words, gene expression of the LDL receptor gene is regulated. So that mRNA levels are changed, so the protein levels are changed. So here's how it works. So well, let's say let's say a cell has a lot of cholesterol already. Intracellular cholesterol concentrations are are high. Intracellular cholesterol concentrations are high. Is the cell going to upregulate or downregulate receptors? Downregulate. So if intracellular cholesterol is high, then it downregulates LDL receptors. Right? It doesn't need more cholesterol. And if intracellular cholesterol concentration is low, what does it do? Upregulates LDL receptors. What about blood cholesterol? What if blood cholesterol is high? What happens to LDL receptor numbers? Not regulated by that. Not regulated by blood cholesterol. Intracellular cholesterol. That's a really important principle of this. So what these guys found out, Brown and Goldstein, they actually followed this. I think they first characterized this like, you guys, your parents hadn't even met by then. This was like mid-80s. The 80s were fun, believe me. And one of the things that they were doing in the 80s was they figured out this process of how the LDL receptors are involved in all this, and then they kept following. And I've got papers like all the way through through the 90s, and about 2000, early 2000s is when they went, there's a protein inside cells that sits inside the RER membrane that basically senses cholesterol concentrations in the cell. And for example, when that cholesterol concentration drops, this protein breaks away from the RER, goes into the nucleus, interacts with other proteins, and changes gene expression of the LDL receptor. So they characterize that protein and all of this stuff. So that's what is driving all this, intracellular cholesterol concentrations. Now, let's go kind of take a step outward and go, okay, well, how does this affect a person's blood cholesterol levels, and how does that relate to heart disease? One of the things we talked about in the fiber lecture is, we actually talked about this before when we talked about digestive system. We said, if a person is recycling bile acids, right? The bile acids are made from cholesterol in the liver. Say this is a bile acid, comes from cholesterol. Remember that bile acid goes from the liver. Bile acid goes into the gut, participates in the emulsification process, the mixed micelle formation, and then remember that bile acid is picked back up, leaves the ileum, goes back to the liver. And one of the things we said was, well, cholestyramine stops that process. It binds bile acids in the gut. So does fiber. Certain types of fiber bind bile acids and cholesterol in the gut, and they eliminate those bile acids or cholesterol. So now what happens to this recycling? It stops. So this cholesterol molecule that was just made from acetyl-CoA would go to bile acids instead of going into VLDL. So if you're reducing recycling of bile acids, you're decreasing the concentration of liver cholesterol. So what does that do to LDL receptor numbers? Increases it. Let me run through that again. So for example, if a person is consuming fiber, some fiber binds bile acids and cholesterol in the gut. That decreases bile acid recycling. And that decreases hepatic cholesterol concentrations. Let's say this bile acid is going down the small intestine. And as this bile acid goes down to the small intestine, it binds fiber. This bile acid, along with fiber, is excreted. That means that there's less recycling. That means that this cholesterol molecule, rather than doing this, does this. More cholesterol is now becoming bile acids instead of remaining as cholesterol. So that decreases hepatic cholesterol levels. So what happens to the LDL receptors? They increase in number because intracellular cholesterol concentrations have gone down. So now what happens to the LDL in the blood? It's taken up by the liver. It doesn't participate in atherosclerosis because it's taken up by the liver. So what are you doing? It's decreasing blood LDL cholesterol concentrations by consuming fiber. And the mechanism is through the LDL receptor. Let me give you a simpler example so you can make sure that clicks for you. Another example is, have you ever heard of statins? What do statins do? What are they?
Drugs you for prescription drugs that you take for if you have high levels of LDL cholesterol.
They're medications that can decrease blood cholesterol. One of the most popular common drugs ever. They're multi-billion-dollar drugs. And other names are like Lipitor, Crestor, what else? Atorvastatin. There's a few of them.
Aren't those the medications that like block the reuptake in the ileum?
Now these guys, statins, decrease cholesterol synthesis by decreasing HMG-CoA reductase activity. Remember that one? The rate-limiting enzyme in cholesterol synthesis. And what's the most important tissue for cholesterol synthesis in the body? The liver. So these drugs can decrease cholesterol synthesis in the liver, which means intracellular cholesterol concentrations in liver cells goes down. So what do the cells do? Make more receptors. So those drugs are working through this pathway that Brown and Goldstein figured out. They basically realized if you control intracellular cholesterol concentrations, you control LDL receptor numbers. And that basically, those drugs essentially draw cholesterol out of the blood into the liver. And then the cholesterol can be metabolized to other molecules that aren't harmful. So that's how those drugs work. That's how things like fiber work. and some other things that are in the diet that bind cholesterol. Got all that? And you'll see in the extra credit assignment that's Lipid Lecture 3, there's a short little section in there that talks about regulation of LDL cholesterol, and it will mention things like saturated fats also tend to regulate blood cholesterol through the LDL receptor mechanism. So it's important that you can go through that. Good there? Okay. Let's go to the good guy, the HDL cholesterol. So HDL particles, right? They perform what's known as reverse cholesterol transport. So it's considered the good cholesterol of the plasma because HDL is taking cholesterol from extrahepatic tissues to the liver for excretion. And they have a protein called ApoA1, and that's crucial for HDL structure, and also activates an enzyme called LCAT. And the cholesterol ester in HDL can be transported to the liver for excretion. I'll go through that process right here. So here's kind of an overview of what it looks like. I tried to clean this up in some program. Hopefully it looks okay on your printouts. So blood, obviously, endothelial cells, underlying cells, including these. FC is free cholesterol. So here's the way HDL work. When HDL are made, they're kind of flat little discs, and they don't have much on the inside. They're mostly just phospholipids. And because the phospholipids, because there's so few of them, they basically just form this almost flat particle. Okay? They're really small and dense. They're so small they easily leave the blood, go between the endothelial cells, and interact with the surface of underlying cells. They pick up free cholesterol. So HDL leave the blood, scavenge cholesterol, and basically incorporate it. So they're picking up cholesterol. As they get bigger, what do they need more of? Phospholipids. So PL is phospholipids. So they're bringing in cholesterol. They're kind of mixing it in with their phospholipids. They're taking phospholipids from the surface of cells. They do that until they get to a certain size, and then they kind of go through the interstitial fluid, the lymph, and go back into the blood. In the blood, HDL interact with LCAT. So see this little thing here? That's HDL plus LCAT. So LCAT is an enzyme that converts cholesterol to cholesterol ester. I'll show you the reaction in a second. So LCAT converts cholesterol to cholesterol ester. And I'll show you, let me, if I remember where this is. So think about what's happening here. This HDL has picked up cholesterol. Now, one of the things I didn't tell you before was if lipoproteins in the blood interact with each other, that's relatively common. They can exchange apolipoproteins, they can also exchange cholesterol. So if this HDL is loaded with cholesterol and it's interacting with other molecules or lipoproteins or other tissues, some of that cholesterol can come out. So what HDL do is they take cholesterol, convert it to cholesterol ester. So it goes from the surface to the interior. Now which enzyme does that? LCAT. So LCAT converts this cholesterol to cholesterol ester. And that almost guarantees the cholesterol is going to go to the liver. So LCAT converts cholesterol to cholesterol ester for transport to the liver. So this kind of describes it and shows a little bit of it. Here's cholesterol. Here is phosphatidylcholine, lecithin, very common phospholipid. Very common phospholipid on the surface of HDL. So what this, and on the surface of cells. So here's cholesterol. This fatty acid from lecithin, so remember this is a phospholipid, got fatty acid right here. LCAT takes this fatty acid and adds it to cholesterol. So you end up with a cholesterol ester and a lysolecithin. So the name of LCAT, so LCAT stands for lecithin cholesterol acetyltransferase, which basically means it takes a fatty acid from lecithin, transfers it onto cholesterol. So which apolipoprotein activates LCAT? A1. ApoA1 activates LCAT. That's why it's there. It's the main reason it's there. Got that? So again, big picture, think about what HDL is doing. HDL is made in the liver, it's made in the small intestine, goes into the blood, leaves the blood, picks up cholesterol, goes back to the blood, allows LCAT to convert cholesterol to cholesterol ester, and then HDL goes to the liver and is absorbed. So that's what this one shows. Here's HDL. It's picked up cholesterol, converted it to cholesterol ester, takes it to the liver. That's going to be excreted, more than likely. How's it going to be excreted? Yeah, in bile, so the cholesterol can go into bile, or the cholesterol can be converted to bile acids. So once that cholesterol from HDL goes to the liver, it's either going to be converted or it's going to be excreted in bile. So that's how it's getting rid of it. That's why that bile system's so important. It also helps us regulate cholesterol levels. Right? So now think about it. Why is it important to have high HDL in the blood? Because if you measure that, and what you'll do in 329 is you'll get your blood sample, and then I'll give you a reagent, and you'll use that to spin your plasma sample, and it will precipitate LDL, and HDL will stay in solution. So then you'll take a sample of that, and you'll measure it, and you'll say, oh, my HDL cholesterol is 68 mg per deciliter. That means that cholesterol was cholesterol in your blood that was on its way to the liver for excretion. That's why it's good to have higher amounts of that, because you know it's basically on its way to be excreted. Got all that? How can you?
Once it's in your blood, it's on its way to be excreted?
If it's HDL, yep. Yep.
So how do you increase HDL levels?
Not easy. Not easy for anybody. It's harder for guys. There's a few different ways. Have you heard that? Like red wine is good for cholesterol. Never mind then. So things like moderate alcohol consumption can increase HDL cholesterol. Okay. Is it just red wine? Alcohol is part of red wine. I should say yes, especially because of where we live. We live in wine country, right? No, alcohol in general. Red wine also has the additional benefit of antioxidants and other helpful molecules like resveratrol and things like that are also components of that. So it has that going as well. Is it a really significant effect? It is not significant enough for a person to start drinking. Okay? So keep that in mind. What else? Certain types of exercise can help that as well, can increase HDL cholesterol. And the types of exercise, a variety. It needs to be intense, rigorous exercise repeatedly in order to affect that. It's not that easy, but there are some things that you can do to help that. Good there? Uh-huh.
I think it's easier to lower LDL than to increase HDL.
Yes. Yep. Quite a few things that can decrease LDL cholesterol. Again, diet, exercise, things like that. Insulin sensitivity affects it. Omega-3 fatty acids, and you'll see in the lecture that there's a few things that do that. But yeah, it is in general easier to drop LDL cholesterol than increase HDL. And sometimes there's a trade-off. Sometimes there's not. I mean, sometimes if LDL is going down, HDL is going up. But a lot of times you'll settle for a drop in LDL. Uh-huh.
When you say insulin sensitivity, do you mean just like that period after you work out, or in general? Is there a
No,
way
that
to-
means like one of the downsides of if a person is adding adipose tissue, and insulin sensitivity is insulin resistance is changing in some tissues. Sometimes that insulin is still able to increase HMG-CoA reductase. And in general, if the liver's making a lot of cholesterol, it's going to put it into VLDL. And in general, the more VLDL you have, the more LDL you have, because that's where it comes from. So, but that also has to do with some other factors as well. Let's see if I can get the next one on here. So the next one that we're going to talk about is Lipid Lecture 4, because Lipid Lecture 3 is the extra credit. And that's about a 43-minute video, I think. Yep.
What classifies it as HDL or LDL?
What classifies?
Like, what's the difference between HDL and LDL? As far as like, if you were to test for it, like, how can you tell which one's which?
So the way we're going to do it is we're going to take advantage of the size and buoyancy difference. So LDL, because it has ApoB100, precipitates pretty easily. So you guys are going to use a compound called polyethylene glycol that I'll give you in a little buffer, and you're going to vortex it and mix it, and that will decrease the hydrogen bonding between the LDL ApoB100 and the water in the plasma, and it'll cause it to drop. And then you're going to spin it. So you're going to distinguish it just by the HDL will stay up. It'll be in the supernatant. In the lab, what we used to do is you float them up separately, like I talked about before. You use the density differences in the lipoproteins. And HDL is so dense that you just use the difference in the gradient in your tube when you spin it. Yeah. That's typically the way you do it clinically. The way we used to test the fractions is after we did a separation, we'd run a protein gel. And then we could look at our proteins, the apolipoproteins on there, and tell how clean our separation was. So, for example, if we had chylomicrons and we saw B100, we had a problem. So, good there?
What does IDL stand for?
Intermediate. IDL is intermediate density lipoprotein. Good catch, I didn't say that. Or Vldl remnant is the same as intermediate density. Lipid Lecture 4, I propose we talk about burning fat. Okay, so here we go. This is the outline of what we're going to talk about. Catabolism of triglycerides, of fatty acids by an enzyme called hormone-sensitive lipase. And then essentially what we're going to do is we're going to kind of follow those fatty acids and see why they're such a good source of energy. So we're going to talk about fatty acid oxidation. We'll do some number crunching, ATP and all that. And then we'll talk about ketone bodies. We'll talk about how carbohydrate limitations cause ketone body production. We'll look at specific reactions and we'll talk about the effects of ketogenesis. And then we'll talk about fatty acids and triglyceride synthesis. So in this case, we'll kind of go from starvation, limiting to the excess, and we'll talk about how we make triglycerides and how they're stored. And that'll wrap up lipids. And then we'll go into proteins. Okay? So let's start off by talking about this enzyme. This enzyme is called hormone-sensitive lipase. And as the name implies, it's regulated by hormones. And let's see. This is an adipocyte. So here's the blood. This is a triglyceride droplet in a fat cell. So hormone-sensitive lipase actually breaks the triglycerides into fatty acids and glycerol. So it catalyzes the release of fatty acids from the stored triglyceride droplets in adipocytes. There's also some triglycerides in muscle. Most triglyceride is stored in fat cells, but there's also some in muscle. That's mostly where we get it, is from the fat cells. And it's kind of counterintuitive. Actually, as a person becomes more advanced in their cardiovascular condition and training, muscle cells get better at storing triglycerides. And it's kind of a little bit of an additional fuel source for them. And so hormone-sensitive lipase is also activated in muscle fibers. But let's talk a little bit more about fat cells or adipocytes. Their major function is to store triglyceride. And it's not uncommon for 80 or 90% of the volume of an adipocyte to be triglyceride. So they'll actually continue to add triglycerides to this droplet. And you see there's images that actually show all the organelles and everything is basically pushed to the side as this triglyceride droplet kind of dominates this fat cell. And then when that triglyceride droplet is hydrolyzed, it shrinks in size. And then when triglyceride is added, it expands again. So fat cells have a impressive ability to change their size or volume. And you guys have all probably heard things like them saying, well, you know, once you have a fat cell, it stays around. You can't get rid of it. Have you ever heard that? There's some truth to that. They're not always easy to get rid of. They can go away, but their kind of ability has to do with them being able to expand and shrink in size. And essentially, sometimes they're kind of waiting for me to eat too many calories so that now it can expand its volume again. It's kind of what they do. There's certain times in life where we add more fat cells more readily. And one of them is basically through adolescence, but also earlier in life. So if a child, for example, is consuming more calories than they're expending and they're adding adipose tissue, sometimes it creates a challenge for them to get rid of those fat cells as they get older and develop. It absolutely is more challenging. You've probably heard some people say, man, all I have to do is walk by a place that has good food and I gain weight. There's a little bit to that. If those fat cells are basically shrunk in size and they're kind of waiting for additional calories and triglycerides to come into play, they can easily expand. So we have to be careful with kids as they go through development. It's really important that we try to maximize their health benefits for the rest of their life by doing things like keeping them in good shape when they're growing. And I think Dr. Adams alluded to, man, it's easy not to exercise anymore. It's easy not to do physical activity. You guys are, I mean, when I was growing up, my mom and dad basically kicked us outside. That was it. Why are you inside? TV goes off, boom, out. You're outside all day. That's all we did. Different, a lot of things were different, but we didn't watch stuff. We didn't have video games or anything. I was terrible at video games. That's probably why I didn't play them very often. To be fair to myself, I wasn't coordinated at them. But that's kind of what we have to keep in mind is kind of the physiology of these fat cells. And can we get rid of them over a period of time? Yeah, some of them will go away. Some of them will go through apoptosis and get rid of themselves. But one of the things that they realized a few years ago, they used to say, well, during development and everything, you can actually increase fat cell numbers. But then adults, you basically have the number of fat cells that you're going to have, and you just expand or decrease their size. Is that true? A few years ago, they realized, wow, almost at any point in your life, you can increase numbers. Isn't that great? Right? So there are certain times in life where they're harder to add or to lose, but we have to keep in mind that that's their main job, is to stay around and adapt to the amount of triglycerides that are present. So here's kind of how they work. They are a storage location for energy that can be used in any part of the body. Sorry, almost any part of the body. So the major function of fat cells is to store energy that can be used in other parts of the body. Fat cells are not very metabolically active. They don't need a lot of energy. So they provide energy to other cell types, like muscle. I think I mentioned that before, but you've heard that. Have you ever heard that phrase, a resting muscle cell burns fat? That's true. So in general, we say, well, if you can stimulate muscle fibers to require energy, even when you're resting, you're kind of burning fat. Then that kind of helps a person maintain a healthy body weight. So if we look at what's happening is, under non-stimulating conditions, hormone-sensitive lipase is in the cytosol. That's the black circle. When hormone-sensitive lipase is activated, it moves to the surface of the triglyceride droplet. It's activated by phosphorylation. That's what that P means. So when hormone-sensitive lipase is activated by phosphorylation, it moves to the surface of the triglyceride droplet. And then what does it do? Converts triglyceride to free fatty acids and glycerol. And the free fatty acids run down their concentration gradient, right? Imagine them accumulating in the cell. They'll run down their gradient out of the cell. When they go into the blood, they're hydrophobic. They'll look for pockets on albumin to bind. And that's how they're transported through the blood. Right, so they can go to all different tissues. Like if I'm storing fat around my abdomen, that fat can be used by other muscle fibers in other parts of the body that are that require the energy. So we're essentially going to kind of track these fatty acids. I think I went through all that. And another fascinating area of research is, you know, this just shows kind of a triglyceride droplet. It's actually surrounded by a family of proteins called paralipins and other proteins that kind of regulate fatty acids coming in and out of those triglyceride droplets. So there's several different layers of regulation in here that we're not going to talk about, but that can actually affect this process. All right, hormone-sensitive lipase. So I don't know what made me think of this, but if any of you guys are around next year is when we're going to again offer the series of metabolism advanced classes. So like there's a FSN 528. I don't know if I'll teach it or someone else will teach it. I taught it last year, but it's kind of the next step for 328. So we go into more advanced topics like I talk about ketogenesis a lot more, how all this fat oxidation is regulated, metabolic flexibility, all of that. So it's a 4-unit class. You guys would be welcome if I teach it to either sit in or take it for credit if you want to do that. And then there's a 2-unit graduate class on vitamins, and there's a 2-unit graduate class on minerals if you guys want to take any of those. I'm sure there'll be a long line out the door anyway. So here is the regulation of it. In the fed state, typically when we consume mixed meals, insulin goes up. And I've said this a few times, but if glucose is available, it's the preferred energy source. So in general, when blood glucose goes up, fat oxidation goes down. And one of the reasons is insulin decreases hormone-sensitive lipase activity. That's one of the reasons. You might have heard that. Exercise in the morning before you eat any carbohydrate because you'll actually burn more fat. You heard that? Because insulin stops fat oxidation. Is that true? A little bit. But I would say eat before you exercise for a lot of other reasons other than that. But that's what they're driving at when they want you to, you know, curb your body's cravings for carbohydrate. They'll say stuff like that. I don't know who they is. Lots of people, I guess. Here we have fasting or exercise. So when you're fasting or exercising, stored triglycerides used as a major energy source. And one of the reasons is because glucagon and epinephrine levels go up in the blood and they activate hormone-sensitive lipase. And therefore, in times of lower blood glucose or exercise, free fatty acids serve as a major energy source for tissues absorbing them from the bloodstream. And here's how it works. Imagine epinephrine, right? Physical activity, epinephrine goes up. It interacts with receptors on the surface of fat cells. Signal transduction causes phosphorylation of the enzyme. The enzyme moves from the cytosol, the triglyceride droplet hydrolyzes triglycerides. Glucagon is going to be stimulated when blood glucose goes down. So epinephrine is always going to go up in physical activity, or at least it should. Glucagon sometimes takes a little bit of time because it's not regulated on physical activity, it's blood glucose. So blood glucose drops, and typically during exercise, blood glucose is down and glucagon is up. And during that scenario, insulin is usually relatively stable. Sometimes insulin will go up and down a little bit, but this would be insulin. Insulin binding its receptor when blood glucose goes up and insulin is released, inactivating hormone-sensitive lipase. So it's facilitating glucose use for energy. And then when the fatty acids are hydrolyzed, they're bound to albumin in the blood. Next class, I'll start off by giving you an example of those hormones and how they change during an exercise regimen. Any questions? Okay, guys, thank you. Have a good night. I'll see you Wednesday.